First we will be learning about mastication. So we will be taking 2 crackers and chewing them for 2 minutes without swallowing and setting a timer and then answering questions after. For the question, so what physical changes did you notice about the crackers as you chewed? So it became like very broken down and it just like turned into nothing. Like it was just mush. What chemical changes in taste did you notice? So at first it was like a little salty, and then there was like no taste. It literally tasted like nothing. And then how would you describe the taste over time as you were chewing? So at first it was salty, and then it just like tasted like nothing. It was just. So next we'll be learning about muscular action in your stomach. So we have a bag with with muscular action and with no muscular action. So we'll add 1 cracker and 1 cup of vinegar to each bag and record what it looks like initially. Okay, so I just put the crackers and the vinegar in there. So initially it just looks like a cracker with vinegar. Okay, so the bag with muscular action, and I squeezed it 6 times. You can see the cracker is dissolving and very flimsy. The one that's just sitting here is flimsy, but it's not like falling apart. So now we'll do it again for another 5 minutes. Okay, so after 10 minutes, see the cracker is now really starting to fall apart. And this one's still sitting still. So now I squeeze this one 6 times again. And go another 5 minutes. Last 5-minute timer went off, so here's what the mastication one looks like. Or with the muscle action. Here's what with none of it looks like. So it's definitely flimsy, but it's not falling apart after I squeezed it 6 times each time. So now we will answer the following. Okay, so the first question is, why did we add vinegar to the bags? Because the vinegar helps break down the food and made it easier for our body to digest. What difference did you see between the 2 bags? So the one with mastication, so when I squeezed it 6 times each time, it helped break it down. And then this one is still whole, but breaking down slowly. Which bag did a better job at digesting the crackers? Why? Definitely this one, because of the muscle contractions of squeezing it. It helped break it down into smaller pieces. Okay, so now we're going to learn the curvatures of the small intestine. So we will get 4 paper towels, 2 glasses with 2 cups of water in each one. Okay. Fold 1 paper towel into 4ths and place it in 1 glass. Okay. So fold 1 into 4ths and fold the other 3 paper towels into 4ths. Okay, set a timer for 30 seconds, and at the end of the 30 seconds, remove each paper towel. Okay, so now we pull the paper towels out without squeezing them and throw them in the trash. And now we will answer some questions about it. First, I'll show you how much water remains in each cup. So you can tell the one that had 3 paper towels definitely soaked up more. So the first question is, which paper towel treatment had the largest surface area? And I think that would be the one that had 3 paper towels, because there was 3 whole paper towels instead of 1. Which treatment had the least amount of water remaining? The one that had the 3 paper towels had the least water remaining because there was 3 paper towels soaking instead of just 1. Which treatment would represent the best version of how our small intestine absorbs nutrients? I think the one that had the 3 paper towels in it, because our small intestines are so long, so that it's absorbing like constantly as our food is moving through it. We'll be going over the teeth. So the first question is, what is the lower jaw called? And that is the mandible. And your lower jaw is your mandible, and then your upper jaw is your maxilla. And then how many central incisors do we have, and what is the function? There's 4, and they're mainly used for cutting food. How many lateral incisors do we have? And we have 4, and they're also used for cutting food. How many canines do we have, and what is their function? We have 4, and they're for gripping and pulling. How many premolars do we have, and what is their function? We have 8, and they are used for grinding down food. How many molars do we have, and what is their function? We have 12, and they are for grinding down food. Why is it only 28% of people have normal set of teeth? And that is because they keep their wisdom teeth. What are your third molars called? And those are also your wisdom teeth. Your third and fourth are your wisdom teeth. Why do people get their molars? Why do people get their third molars removed? Because you don't have enough room. Some people don't have enough room, and they're supposed to go straight up through the hole, but oftentimes they go sideways, and then they cram all of your teeth. What are your gums also called? And it is gingiva. And then here is the diagram. So you can see your incisors, and then your canines, then your premolars, and then your 3 molars. I will label my teeth. So here are my central incisors, lateral incisors, canines, premolars, and then molars. And my wisdom teeth are back here and here. I have not gotten mine out, so I still have all mine. And then your mandible is your bottom jaw, and your maxilla is your top jaw, and then your gums are your gingiva. The next one is learning about salivary glands. So obtain 2 suckers, glass of water, and then place 1 sucker in the glass of water and 1 in my mouth. And then we'll start a timer and see which one can dissolve faster. Okay, we're 2 minutes in. It looks like that. This is what the one in the water looks like. Now we're 5 minutes in. Okay, so at 6 minutes 32 seconds, the one in my mouth fully dissolved. And we are now at 30 minutes and 49 seconds, and just little tiny pieces at the bottom. So 30 minutes and 49 seconds in the glass of water. So which treatment resulted in the sucker dissolving the fastest? The one in my mouth definitely dissolved way faster. And why do we think that was? Because I was constantly salivating. Yeah, I was constantly producing saliva to break it down into smaller pieces and dissolve it. And this one was just sitting in water. There was no way for it to move around. Okay, the last one is determining the length of my alimentary canal. So I will take this string and measure all the different pieces, and then color it different colors so we can see the difference, and then put it all together. So for my mouth, we'll take a piece of yarn, runs from the back of my ears to the front of my lips. So that much. Next we'll measure my esophagus to the bottom of my chin, to the part inferior to my sternum. So right here. Okay, so next we'll measure for my stomach. So from my pinky to my thumb, spread out. Just to about right there. And then next, to obtain the length of my small intestine, I'll take a piece of yarn that runs from the top of the superior part of my head to my feet 4 times. And this is how long my small intestine is. So we ended with my mouth being 7 1/4 inches long. My esophagus was 11.5. My stomach was 8 1/4. My small intestine was 266. And my large intestine was 66.5, totaling for 351 1/4 inches. Okay, post-lab questions. Number 1, what is the function of the digestive system? So it is to break down food into smaller pieces and break it down. Compare mechanical and chemical digestion. So both break down food into smaller parts. But mechanical is the physical breakdown of the food done by the teeth, and chemical is the enzymatic reaction that breaks down the food. What is the pathway of the alimentary canal? So mouth, pharynx, esophagus, stomach, small intestine, large intestine, rectum, anus.